Hi, I'm Nancy Maisels. I'm a professor in the departments of immunology and biochemistry at the University of Washington Medical School, and I'm also director of our molecular medicine program. This is a program that's designed to bring elements of medical training to our PhD graduate students, and we've been lucky to have several hundred graduate students in our classes and over 20 students enrolled in our certificate program from all the basic science departments and the MCB graduate program at the medical school. We're really lucky to have funding for this program from the Howard Hughes Medical Institute, HHMI, and HHMI has graciously given us funds so that we can have public lectures here in Kane Hall at the University of Washington and record them for rebroadcast on UWTV and the Research Channel. So I'd like to welcome everybody who's here in the audience tonight, as well as people we anticipate will be watching at home and on the gym and anywhere else they find comfortable. I'd especially like to welcome all the students we have here in the audience tonight from Nathan Hale High School, who came and had dinner with our students and Dr. Overbaugh before the lecture. So if you guys want to wave, great. Thank you for coming. We're really glad to see all of your faces. Terrific. So it's a real pleasure to have Julie Overbaugh as our speaker tonight. Julie has worked for many years to understand both the mechanisms of pathogenesis and transmission of HIV. And she's focused on transmission to women and transmission from mothers to their infants. So this is an incredibly important subject. And she's developed lots and lots of contacts in Kenya, where she travels regularly, and has been able to bring Kenyan scientists to her lab too. So she's an important connection between Seattle and other parts of the world, as well as a wonderful contributor, not only to research, but to our entire community. She's a wonderful citizen and a great teacher. And Julie was raised in Connecticut and went to the University of Connecticut, where she almost had another career. She played basketball for what's now the best women's basketball team in the nation, but she forsook that to do scientific research, and I think we're all lucky that she did, at least to us. She went on after she finished college to graduate school in Colorado. She went for a postdoctoral stint at Harvard, and then she came to Seattle, and she's been here at the University of Washington and the Hutch for about the last 20 years. And Julie has received lots and lots of recognition for her research contributions, and she seems to be especially proud of her recognition for her contributions as a mentor of students both from here and from abroad. And it's an absolute pleasure that Julie has agreed to speak to us tonight, and here she is. Thank you for being here.
Thank you, Nancy, but I feel I have to set the record straight. I was never really good enough to play basketball beyond where I did, so it wasn't necessarily a choice, but thank you for that. And thank you all for coming tonight to allow me to tell you about some of the studies of our Seattle-Kenya collaborative team. This involves investigators at my lab at the Fred Hutchinson Cancer Research Center, as well as numerous groups at the University of Washington, and our collaborating institutions in Kenya, the University of Nairobi, and Coast Provincial General Hospital. And these are collaborations that, as Nancy said, are focused on trying to develop a better understanding of how HIV is transmitted and how it causes disease. I don't think I need to spend a lot of time dwelling on why we're studying HIV and trying to prevent HIV infections, but this slide sets the stage. These estimates from the UNAIDS website suggest that about 33 million people are currently infected with HIV globally. As you can see in this slide, the majority of affected individuals are in developing countries, particularly in sub-Saharan Africa as well as parts of Asia. Because HIV is as yet not a curable disease, and because healthcare in these settings is sometimes weak, this presents an enormous burden for people in these countries. Importantly, from the point of view of efforts to block HIV transmission, there's an estimated 2.5 million individuals who have been infected in each of the last several years. Most of these individuals are infected through sexual transmission, but blood exchange during injection drug use and transmission from a mother to her infant in utero during delivery and during breastfeeding all contribute to these numbers. So of course, this spurs a lot of us on to think about ways to reduce the risk of HIV acquisition. And I kind of categorize the approaches that are currently being taken or being developed into two general categories. One is an effort to reduce risk, and the others are efforts that are directly targeting the virus. And I'll talk about both of those for a few minutes, but I'm going to talk today about efforts to directly target the virus. But certainly reducing risk has been an important way in some populations to impact the spread of HIV, and particularly behavioral changes that reduce the exposure to HIV by reducing high-risk encounters, or methods such as using condoms, barrier methods that prevent acquisition. More recently, it's been appreciated that male circumcision is actually an effective way to reduce transmission of HIV because a number of trials have shown that circumcision of men reduces the risk of their HIV infection. More recent studies have focused on other ways to try to prevent HIV indirectly, for example, by treating cofactors that are known to increase risk of HIV acquisition. And one such cofactor is herpes simplex virus type 2. There are other sexually transmitted diseases that, if present at the time of exposure, appear to increase risk of HIV acquisition. And so a number of trials, including a large trial led by investigators at the University of Washington, are focused on targeting these cofactors as an indirect means to reduce the risk of HIV acquisition. And those approaches remain to be proven. It'll probably be another year or two before we really know if targeting HSV2, for example, is effective. In the category of directly targeting HIV itself, there have been a number of attempts to do this by providing microbicides, which are female-controlled topical methods designed to block or inactivate virus during sexual exposure. A number of compounds have been tested as microbicides, and none have as yet been shown to be effective, but very recent data put out about a month ago suggests that there may be one compound that's beginning to show promise as a microbicide. So this continues to be an active area of HIV prevention research. Another area that's been the focus lately is an approach called pre-exposure prophylaxis. And this is kind of akin to what we do with malaria, where if you're going to a region where you expect that you might be exposed to malaria, you take anti-malarial drugs before you get there. And the point of this approach for HIV is really to do the same thing. And one of the great successes so far in HIV research has been providing pre-exposure prophylaxis to infants of HIV-positive mothers. And this has significantly reduced the risk of infant infection in this setting. Now, the drugs are provided to both the mother and the infant, so it's a little unclear at this point whether the drugs are working by reducing the virus in the mother or by providing prophylaxis in the infants. But just a single drug given to a mother at the time of delivery and a single drug given to an infant at the time of birth can reduce the risk of HIV acquisition by almost half. So this approach has spawned the idea that we might use this in adult populations who are exposed through sexual contact or through injection drug use. And there are a number of trials that are just starting to test this concept. I think it'll probably be a few years before we know the results of those trials. But the holy grail of HIV efforts for prevention has really been to try to develop an effective HIV vaccine. And this is the desired approach because if we can develop an effective HIV vaccine, we should be able to elicit durable immunity to this infectious agent. So what I want to talk about today specifically are thinking about, in designing things like vaccines and other prevention efforts that are targeted at the virus, what viruses are we actually targeting? And as I show you in the next couple slides, HIV is a very diverse virus. It's genetically variable. It mutates rapidly. And so this presents unique challenges to developing HIV vaccines. I want to spend a lot of the rest of the talk talking about immune mechanisms and the possible role of neutralizing antibodies, specifically in protection from HIV. And then at the end, I'll just share some of my thoughts on whether antibodies are actually the way forward in HIV vaccine design. Let me start with going through the key steps in HIV replication, because this is really at the heart of what makes HIV so difficult to block. So viruses like HIV really have to do three things to be successful. They have to enter the host cell, they have to replicate their genome, and then they have to produce new virus particles and send them out of the cell. In the case of HIV, entry of the virus is determined by an interaction of the envelope protein, shown here in the little blue balls in the upper left corner, and it binding to a receptor on the host cell called CD4. A second receptor is required. This is a chemokine co-receptor, and it can be one of several chemokine receptors. It's most often a protein called CCR5. The cells that express CD4 and CCR5 are T lymphocytes, macrophages, and dendritic cells, all cells that are key parts of the immune system. And in fact, it is infection of T lymphocytes that is responsible for the consequences of HIV infection, which are immunodeficiency, because the virus destroys these target cells. Once the virus enters a cell that has the right receptor and co-receptor, it then has to replicate its genome. And HIV is a type of virus that's called a retrovirus, and it's called a retrovirus because it reverses the flow of genetic information. HIV is an RNA virus that goes through a DNA intermediate, and our host cell usually does the opposite. It starts with a DNA form and generates an RNA form. As you can see in this slide, when the DNA copy is generated, it's then translocated into the nucleus of the cell, and it integrates into the host genetic information. Once this form is integrated into the host cell, it becomes a permanent part of the genetic code of that particular cell. So at present, the only way to get rid of HIV once the cell's infected is to get rid of the infected cell, which again makes this a very complicated virus to try to target. Once it becomes a DNA form in the cell, it basically just takes advantage of all the machinery the host cell has to offer to make more RNA, to make more proteins, and to generate copies of itself that can go on to repeat this cycle. One of the characteristics of HIV replication that makes this virus so tricky is that during the process of making a DNA copy from its RNA, it often makes mutations. This process is carried out by a viral enzyme, reverse transcriptase, and it doesn't go back and correct its mutations, it just lets them go. And so as this schematic shows you, in the DNA form of the virus, there's often a mutation, shown as the red flag here, that's fixed in the DNA form of the virus. And because that DNA form of the virus becomes the template for new virus particles, the viruses that leave the cell are often genetically distinct from the viruses that enter the cell. To put this in a slightly different perspective, for those of you who think about mutations that occur between individual people, for example, in the context of mutations that might cause cancer or other diseases, if we looked across a stretch of sequences as long as the HIV genome, 10,000 bases, and you look around the room at the people next to you, assuming they're not relatives, you all differ by about 0.1% in your genetic sequences. If you compare two HIVs from different infected individuals, those sequences, as shown on the bottom, may differ by anywhere from 10 to 30% in sequence. So this is an extraordinary amount of genetic diversity that's not seen, for example, when you compare two individuals. So this adaptation and this flexibility, this ability to mutate rapidly, allows HIV to adapt in the face of immune responses and other selective pressures applied by the host. So if the virus didn't mutate so rapidly, the immune system would have a much better chance of actually getting rid of the virus. But since the virus can just keep adapting, keep mutating, it can evade the host immune system. And as a result of that, if you look at a person who's been infected for several years, you see a considerable amount of virus diversity as shown here. And we've estimated that there's on the order of about 0.5% mutations per year in an HIV-infected individual. So given this amount of diversity, one thing that we have been interested in for quite some time is, are all these viruses equally likely to be transmitted? These viruses have been selected in one person because they can evade that person's host defenses. But it's less clear whether all those viruses would be successful at spreading from person to person. And we think this is important because these are the viruses that we really need to block with the vaccine. So we've addressed this by looking at the viruses that are present during what's called primary or acute HIV infection, as shown here. This is the time when virus replication is very robust. There's high levels of virus replication in those first few weeks and months after acquisition that eventually subsides to set point or steady state level. But by looking at the viruses that are present at this period, we thought we could better understand what types of viruses are transmitted. And we found in our studies, and other investigators have had similar findings, and that is that if you look at the viruses that are transmitted, they're actually genetically rather homogeneous in most people. In fact, some of them are almost identical. So this suggests that there is some bottleneck during transmission that either favors those viruses or perhaps just by stochastic processes allows one virus to get through. Now we found that in some women, and particularly women who have other sexually transmitted diseases as co-infections, that we often find more genetic diversity in the viruses that are transmitted. And I'm showing you here some of the numbers that we've seen, up to 6% diversity in viruses early in infection, but that's a rather rare event. And in most cases, as depicted with this schematic, the diversity in the viruses that are transmitted is much less than the diversity of viruses present during a chronic infection. Now, we and others have shown that these viruses do have particular biological characteristics that distinguish them from the other viruses. I won't talk at all about that tonight because we don't really understand what drives that selective pressure at present. So for a vaccine, what we want to do is protect against those viruses that are getting transmitted. And we want to elicit immune responses by vaccination that are poised and ready when the person is exposed to the infectious agent, in this case HIV. But I've told you that HIV is really a moving target because it mutates so rapidly. And there has never been a vaccine to any infectious agent that is this genetically diverse. So this has really led to questions in the field from the beginning of what's the best way to develop a vaccine, what's the best way to immunize against this virus. And the two approaches that have classically been considered are to try to use an immunogen, a virus, an antigen, to elicit neutralizing antibody responses, which are responses that generally target the virus, or to elicit cytotoxic T lymphocyte responses which target the cell. So neutralizing antibodies work by binding to the virus, the envelope protein in the virus, which is important for binding the receptor, and by doing so they effectively block viral entry. So that means that the cell doesn't become infected, it doesn't produce new virus particles. Cytotoxic T lymphocytes operate by targeting the infected cell and lysing it, so that when they do this, they eliminate the infected cell and they thus eliminate the reservoir for production of new virus particles. So both of these strategies, in theory, make a lot of sense in terms of eliciting protective immunity to HIV. Really, in the beginning, the focus was on neutralizing antibodies. So right from the mid-'80s, soon after HIV was discovered, there was considerable effort put on trying to figure out how to elicit neutralizing antibodies to HIV. And part of the rationale for that was simply that almost every vaccine that's been developed that's effective is based on the idea of eliciting antibody responses and works through the action of antibodies. In addition, data in the last decade has shown that passive infusion of HIV neutralizing antibodies in monkeys protects them from a closely related virus, SIV or SHIV. So for the 1990s, really when these challenges became apparent, the emphasis shifted to focusing on a CTL-based vaccine. And the rationale for this approach was first that CTL responses correlate with viral control in HIV infection. And this has been seen in two settings. The first is that people who have more robust CTL responses tend to have less virus in their circulation. And the second thing is that the CTL responses come up as the virus levels are going down during acute infection. And this is coincident with the development of CTL, suggesting perhaps a cause and effect. In addition, host genes involved in determining CTL recognition, these are HLA genes, are linked to viral control and disease outcome in many studies. And then the third reason that really spawned an interest in this approach was that if you took SIV-infected monkeys and you depleted their CTLs, you saw a bounce in virus, again suggesting that CTLs were important in controlling the amount of virus in the system. However, in 2008, there was really a year of reflection in HIV vaccine research because the first large-scale trial based on the concept of eliciting CTL responses by vaccination, this was the STEP trial or the Merck trial that probably many people read about in the news, this trial failed. Now, it's still unclear if the failure of this trial was a failure of the concept of CTL-based vaccines or a failure of the very specific product that was tested, and this is something that's being actively investigated. But I think what this did is made many people look back and start to realize that it was time to think about how much we really did know about mechanisms of immune protection in HIV and whether we really knew enough to be designing vaccines. And again, the data that I showed you showing a correlation between CTL and viral control is only saying in a person who is infected, these CTLs can control that particular strain of virus in that individual. But there really was no evidence that CTL responses could block a new infection. So this really led a lot of people to a back to basics approach and thinking about how to approach vaccines and to focus more on trying to understand how immune mechanisms work in HIV infection. So the studies of our group have been focused in that area for some time. We've been interested, as I said earlier, and as Nancy mentioned, on the role of viral and immune factors in HIV transmission and pathogenesis. We have a particular focus on populations most affected by HIV in developing countries. And I want to share with you some of our studies trying to address whether we can discern a role for antibodies in providing benefit in protecting from HIV infection in such populations. And I think this is quite important because I just mentioned to you that going into the CTL-based vaccine approach, we did not know if CTLs would protect against infection. We knew that they were correlated with viral control. We feel it's important to try and understand whether neutralizing antibodies are associated with protection in HIV infection before we launch a large-scale effort focused on specifically on neutralizing antibodies. And I'll talk about two populations where we studied this. One is infants of HIV-positive mothers, and the other is HIV-positive people who are re-exposed to HIV after their first infection. So the first study that we conducted was in collaboration with Ruth Ndwadi and Joan Kreis, who are shown here. And Ruth and Joan in 1992 did a trial of breastfeeding transmission of HIV. Now, at the time, it was recommended that women in developing countries, if they were HIV positive, breastfeed their infants. And it wasn't known how frequently breastfeeding transmission occurred and what the timing of transmission was. And these investigators were interested in defining the contribution of breastfeeding to infant HIV infection. And what Ruth and her colleagues showed is represented here, comparing the percent of infants infected over the first 2 years of life, comparing infants who formula fed in the blue line with infants who breastfed in the green line. And I think what you can immediately appreciate is that breastfeeding almost doubled the risk of infection in the infants. You may also notice that looking as early as 6 weeks of life in the infant, you can still see an appreciable contribution of transmission through breastfeeding. So this trial allowed us to address the role of antibodies in infant infection because infants who breastfeed in particular are consistently exposed to virus during the breastfeeding period. We know there's lots of virus in breast milk, and we've shown that the amount of virus in breast milk is a predictor of infant infection. So given that, it's actually a bit surprising to me that more infants aren't infected. The upper line on that green graph was just below 40%. So a lot of infants are not infected despite this continued exposure. We know that passive antibodies achieve high levels at birth and are present during the early phases of life in an infant and during this breastfeeding period when they're at risk for HIV. So we ask whether they are helping to protect against infection in this setting. And we did this by first identifying cases of infants who were HIV negative at birth in the study that Ruth and Joan had conducted, and who became HIV infected or detectable HIV infection by 6 weeks of life. We then isolated the viruses from the mom at the time of delivery, which was about the time she probably transmitted. We isolated viruses from the baby as soon as we could detect them. And we found in this setting, as we had found in adults, that only one virus or a subset of viruses are transmitted to the infant, suggesting there was some sort of transmission bottleneck. So the important question to us here, since we were focused on the role of antibodies, was whether or not in the mother's plasma, whether she had antibodies that were capable of recognizing the virus that was transmitted to the infant. So we had hypothesized that in a mother, she would have a collection of viruses that were both resistant and sensitive to her antibodies. And they would be resistant in part because of this rapid mutation. And if these antibodies were playing an important role in selecting during transmission, then we would predict that the virus that was transmitted to the infant would be resistant to maternal neutralizing antibodies. And in fact, that's what we observed, and that data is shown here. On the left in green are all the viruses that we examined from the mothers. We didn't just look at one, we looked at multiple viruses. And the scale is increasing neutralization sensitivity. So the little boxes at the top are cases of viruses that were very sensitive to antibody, and the ones at the bottom are cases where they were resistant to antibody. What you can see in the infants is that the infant viruses cluster at the bottom of the graph, and they tended to be the ones that were resistant to maternal antibody. You don't see any evidence of viruses that were highly sensitive to maternal antibody being transmitted. In other words, going back to the schematic I've been using, if you look at the viruses in the mom, there's a collection of viruses. There are quite a number that are sensitive to her antibodies, but the ones that are transmitted are ones that have escaped this neutralizing antibody pressure. So the fact that the viruses that are sensitive to antibodies are less likely to be transmitted provides some support for a role of neutralizing antibody in reducing transmission of HIV, and particularly those HIV variants that are sensitive. But what's unclear from this study is whether a mother or any individual would ever have antibodies that are broad enough to protect against the whole collection of viruses that's in an individual. There's hope that this might be true because going back to the slide I showed you a few slides prior, again, about 60% of infants don't get infected. So it's possible that among those infants, some are being protected because the mother has very broad and potent neutralizing antibody responses. And we're in the middle of testing this hypothesis with the study, but I don't yet have any data that I can share with you on this particular topic. We also were interested in asking whether there's ever any evidence that neutralizing antibodies protect against infection in the setting of sexual transmission. And so here, as I show you in this cartoon, in an HIV-infected individual, shown on the left, there should be an antibody response to the infecting virus, whereas in an uninfected individual, there obviously are not HIV-specific antibodies. So we wanted to know whether there's a difference in susceptibility between these two individuals if they're exposed to a new virus. If the antibodies are protective, we would predict that a person who's already infected would not be very susceptible to the second virus. But if they're not protective, then it may be that those individuals are equally susceptible as someone who's never seen HIV. So in this setting, what I'm talking about is a term called superinfection, which is a case where there's infection by a second source partner after the first HIV infection is well established. And it was 2002 when we first realized that superinfection was happening, and not before, and that's partly because it's very difficult to detect. So we wanted to ask how common is superinfection, and when, after first infections, does it occur? The reason we wanted to know when it occurs because if the second virus infects early after the first virus, then it may be at a time before the immune responses to the first virus have developed. So I mentioned to you that CTL responses tend to develop as virus goes down. Neutralizing antibody responses develop more slowly. So if an individual was getting infected early, it may not be that there are immune deficits per se, but simply that the first virus had not had time to elicit a good immune response. However, if superinfection occurs later, it actually suggests that the immune response, after it has had time to develop, is still not adequate to protect against the second exposure. So we addressed this question in a cohort of women that we've been following since 1993 in Mombasa, Kenya. These are women who are HIV negative at the time they're enrolled in the study, and they are commercial sex workers, so they continue to be exposed to HIV positive partners. We've enrolled about 2,000 women in this study since 1993, and despite very active guidance on ways to reduce the risk of HIV acquisition, provision of medical care, including treating their co-infections that increase their risk of HIV infection, because these are sex workers and people who are having sex with men who are likely to be HIV infected, we've now seen about 300 women acquire first HIV infection. Many of those women continue to practice sex and have more partners who are HIV positive, so this allows us to address whether or not these women are becoming superinfected. The setting for this study is the Ganjoni Clinic in Mombasa, Kenya. We also have a laboratory a couple miles down the road at the hospital, as shown here. And because of the study design, where women are enrolled when they're HIV negative and are tested regularly, we know pretty precisely the date of their infection, and we can follow them prospectively. So the approach that we used in this study is first to examine the virus present during that early acute phase of infection by analyzing sequences of the virus and looking at its characteristics. We then analyzed sequences about 5 years post-infection, and in women where we saw that these sequences were clearly different, we then looked in between those time points to determine when the second virus was introduced. Now, I should say that one of the challenges in this type of study, and one of the reasons why superinfection has not been well described, is that you have to discriminate this new virus that might differ, as I said, by 10 or 30%, from the virus that's evolving consistently and continually in the person who's already infected, which over a several-year period can achieve a variation of on the order of 5%. So it requires extensive analysis of genetic sequences. And to do this, we analyze multiple sequences at multiple time points, as shown in this slide. This is a case of an individual who we started to follow at 57 days after their first infection. Then we followed for over 3, almost 4 years. And at each of the time points indicated we isolated and characterized the sequences of HIV in this person. And the first 5 time points we looked at, all of the sequences looked like they were related. They looked like what you would expect for de novo variation, and about 40 of the 40 sequences were all highly related. But at 1,000 days, 1,007 days after the first infection, we saw clear evidence of a second virus, and we saw evidence of that virus at the following 2 time points. Now, one thing you might notice if you look at those numbers, those were the number of sequences of each virus type, is that in this case and in many of the cases we've looked at, you actually do see evidence that the two viruses kind of happily coexist once the second infection is established. So it's not as if the second virus is coming in and taking over. In most cases, it's simply coming in and sort of adding on to the burden. Now, we define in a case like this, we define the interval of superinfection as between the two time points where we didn't and did detect the second virus. So in this case, between 714 days and just over 1,000 days after the first infection. And I'm showing you this same data now in a schematic form. So that bar in green represents the interval that I just showed you on the previous slide where this individual became superinfected. Now I'm going to show you the same intervals for 12 cases of superinfection that we identified, as well as the case reports in the literature I'd mentioned. And so here's that data shown together. Now, when you look at this, you do see that there's a clustering of cases of superinfection that occurred in the first year, perhaps a little bit more than you see later. But I should say that part of that is biased by the fact that most studies focused on individuals that had only been infected with the first HIV for a year or two. What I want you to appreciate from this slide is that there are many individuals who acquired their second infection well into the establishment of their first infection, up through 5 years, which was the longest time frame studied. So again, this suggests to us that cellular or CTL responses would have had time to develop in these individuals, as would neutralizing antibodies. So, of course, one interpretation is these individuals portrayed here are the rare people who did not elicit good CTL responses or neutralizing antibody responses. And they're not like the types of people that have been studied in the past. So we wanted to know whether there are immune deficits in these individuals at the time they are superinfected. And in fact, in some of the previous case studies I'd mentioned, people had looked specifically at whether there were deficits in CTL responses in people who became superinfected, and they didn't find any evidence that this was true. Now, there have only been a few case studies of this, so I don't think there's a clear answer on this yet. But we hypothesized that perhaps in the case of neutralizing antibodies, if they were important for blocking transmission, that the superinfected cases shown in the dotted line would be people who just had very poor neutralizing antibody responses. So to address this, we took advantage of having defined that interval of superinfection, so we knew when the second virus was detected. And we went back to the time point prior to that where we had a sample, and we looked at the antibodies in these individuals. And we looked in two ways. We first tested the neutralizing antibody breadth, and we did this by just looking whether the antibodies at that time could neutralize a collection of viruses that had recently been transmitted in Kenya. And then we looked more specifically at whether there were neutralizing antibodies that could detect the superinfecting virus itself. So in essence here, we were asking whether that virus was an escape virus, much like we saw in the mother-infant study. Well, in the first part of the study where we looked, is there a difference in breadth between people who get superinfected and people who don't? The answer was no. So we looked at 6 cases of superinfection and 3 matched controls for each case. And we basically could not discern a difference in the breadth of neutralizing antibody responses in these individuals. There was some good news in some women because in some cases we observed that the antibodies, although they were relatively broad, were not recognizing the superinfecting virus. So this was much like what we saw in the setting of mother-infant transmission. And it suggested that perhaps the virus was getting through because of an absence of an antibody to that specific virus. However, we also did observe in some women that they had antibodies that could specifically recognize the superinfecting virus, suggesting that at least that level of antibody was not protective in that setting. So I think that these data suggest there's really little evidence that neutralizing antibody responses in people who become superinfected are weak or narrow. Now, this is just 6 cases, but in the first 6 cases, we can't see any evidence that they have a deficit in their neutralizing antibody responses. And importantly, as I mentioned, in some cases, there are even antibodies that recognize the second virus. So to us, these data suggest that the bar for eliciting neutralizing antibodies by a vaccine is quite high, higher probably and broader than what we see in a natural HIV infection where the virus is continually replicating and stimulating the immune system. So I think this is quite discouraging in some ways, but I think we still have a lot more work to do on this topic. But I think it feeds into what really has been the ebb and flow of vaccine efforts and strategies in HIV vaccine. I mentioned the beginning really was the heyday of trying to elicit neutralizing antibodies. As it became clear this was going to be hard, people focused to looking at CTLs as the way forward for developing a vaccine. Depending on who you talk to now and how they feel about the results of those trials, some people would continue to urge that we focus on CTLs, and some people are less supportive. But I think that really most people would say we have to be looking at both. What I would say based on the data we've seen coming out over the last few years, some of which I just shared with you, is that I think we need to be thinking about other. I wish I knew what other was. I'm not sure what it is. But I think the way forward may be a vaccine that elicits a combination of responses, perhaps neutralizing antibodies if we can figure out how to make them broad and potent enough, and perhaps CTL. But I think we need to be thinking about other more creative approaches to eliciting responses through vaccination, such as innate factors or antibodies that work through other mechanisms. So I think the lesson for especially the young folks here is we still have enormous challenges in terms of HIV prevention and specifically vaccines, so you have plenty of time to join the effort. Making a vaccine strain that can protect against all the diverse viruses that are circulating is a challenge that we have not yet figured out how to tackle for any infectious agent. HIV is much more variable than flu, for example, so this is a scale of variation that we just have never tackled before in our vaccine efforts. So there's lots to do. I'll leave you with that thought. I just want to again acknowledge the collaborating institutions. I won't mention all the individuals. Personally, I've tried to note the papers that are relevant from those studies so that you can go look them up. But it's been a large collaborative effort. It's been a great experience to work with the Kenya collaborative team, to work with people who are doing clinical trials, and apply basic science to those studies. So I'm happy to take questions after this.
Julie, this may be a very naive question, but with a virus that has so much mutating and so much variability, is there anything to be learned from the regions of the viral RNA or DNA with that time that don't mutate? Are there any areas that are protected that might be the focus for developing some kind of vaccine?
Well, you just pretend to be naive because that's actually, I think, an important part of this. A lot of people have tried to focus, for example, on the region that's absolutely essential for binding CD4 receptor, because you need to bind the receptor to get in the cell, and that region is therefore conserved. But HIV buries that region, so it's not easily accessible to antibodies or other responses. So that's been the complexity, but certainly focusing on conserved regions has been an area of interest.
It seems to me you have two extremes. One is the HIV mutates rapidly and you get viral species that escape antibodies. The other possibility is that an antibody response could never work, because even if you had infect somebody with a single species of virus for which you had already high levels of antibody for that virus, is that enough to prevent HIV from infecting a subset, escape? Is that enough to infect some cells? So if you load a monkey with high levels of an antibody to a particular virus, and then you infect with that virus that is recognized by those antibodies, do you get a complete prevention of infection. And if you don't, then, going for non-mutated regions of the virus will never work, right? Do we know the answer to that question?
We know it a little bit. So in some of those studies that I talked about, people actually took decreasing doses of antibody and asked where you fail to protect. And it's clear that there is a dose effect of the antibody. And if you just put in a little or moderate amounts, you don't infect every animal. You need to put in a lot. So there really is a dose effect that is
So
apparent.
is that a lot achievable in any vaccine? If it's not, then going after non-mutated regions as epitopes will never work, right?
Well, I think there's a couple thoughts there. One is that we don't know now how to elicit a lot of antibody, but the question is, can we learn? And the second thing is, when you take an antibody and match it to the virus, there are some antibodies that bind better than others. So if we could find a way to make an antibody that's actually better and you need less of it, then it might be achievable. And I think people are thinking about that. So if you can target a conserved region, and if you can make an antibody that's very, very potent in its ability to block virus entry, then perhaps this is achievable. But I would not, if I was a betting person, I'm not sure where I'd come down on this in a 10-year time period, to be honest.
So isn't that testable if you load a monkey with such an antibody, the best antibody you have to the best epitope, and then infect the monkey with that single species of virus? It seems to me that's testable, right? And does it work?
And that's, in the studies that I showed you, they used, again, people wanted their experiments to work, so they used a pretty good antibody.
Okay.
And I think the problem is that we have maybe 4 or 5 different antibodies in the HIV field. It's really been an absence of effort until recently to find the right antibodies, and a lot of groups now are looking at that. We're screening individuals in our populations to look for people who seem to have really strong antibodies to find out what they are. And a lot of groups are doing that because I'm not sure we have the right antibodies in our hands right now.
My question is, I appreciate you giving me an idea of what happens in the cellular sense, what's going on with our bodies with the HIV virus and the antibody. But give me a sense of the social piece of what's happening with people, particularly if you have someone who is HIV positive, they're super infected, their immune system is already impacted. How debilitating are they becoming as you're looking at the study and the social support that's going on with how do you manage?
I think I can't really answer a lot about the social support networks because that's not an area I have a lot of expertise. But what I can say that I think is important in your question is trying to understand how superinfection affects a person and trying to educate people who are HIV positive about the risk that they put themselves at for superinfection is quite important. I think what we know now is that in some individuals, it appears that superinfection may accelerate their own disease progression. And so this is a concern. We don't have enough cases that we know if this is common or rare, but I think we need to figure out in people who get superinfected whether this is a detriment to them, whether it's going to impact their treatment. These are all things that have not yet been well answered, but I think are important.
Let's thank Julie again for a lovely lecture.